And welcome. We're ready to get started with our first speaker, Justin Sankey. Welcome to the 2022 Chemistry Senior Experience Seminar Series, last and final week. Allison Flushman is going to be doing the introduction.
Yay! Cool. So when you schedule your senior talk during lab, the other faculty would love to be here, but sadly they are all in labbing right now. So I get to introduce you, which is super exciting because I met Justin now 3 years ago, 3? I don't even know. Maybe. Anyway, it's been super exciting to watch Justin grow as a chemist over the last couple of years. I have the good fortune of having most of y'all when you're juniors and seniors. And Justin, you were one of my favorite thus far because you'll come in and be like, so like, I understand, but I don't think I understand. And then you'll explain it perfectly. And I'm like, dude, you're fine. So I really appreciate the innate curiosity that you have. So go team to that. So Justin's going to go on after Lawrence. He's going to the University of Rhode Island to study oceanography. And we know he will make a splash. I couldn't help it. Go team. Okay. Anyway, so I give you Justin to talk about stuff. Yeah, you can talk about your own topic. Yay.
Cool. Thanks, Allison. Oh.
Turn the volume down a smidge.
Yeah, it's a little loud.
How about that?
Okay. Is that better? Yeah? Awesome. Cool. So yeah, like Allison said, my name's Justin Sankey. I'm going to talk a little bit about the research I did this past summer as part of my NOAA Holling Scholarship. I was working at the NOAA Biogeochemical Fluid Dynamics Laboratory at Princeton, and I was working under the direction of Dr. John Doon there, focusing on building a biogeochemical model of reactive oxygen species in a marine environment and the role they play in the iron cycle. So I'm going to start you off with a little bit of background just to orient you and give you the terminology you're going to need to understand what I'm talking about. So firstly, what is reactive oxygen species, or ROS? So reactive oxygen species are reduced forms of oxygen, such as superoxide, hydrogen peroxide, and hydroxyl radicals. And they really are what ends up controlling our redox potentials within our oceans. So for the purpose of my model, I've really focused on superoxide and hydrogen peroxide, so we can kind of forget about the hydroxyl radicals for now. So there's two main forms of production for reactive oxygen species. It's a light and a dark reaction sequence. So focusing on the light first, we have at the top of our screen our sun coming in, and at A, we have reaction with things like phytoplankton and bacterioplankton to produce reactive oxygen species. And then at B, we have our photolytic reaction sequences happening with CDOM, which is color-dissolved organic matter, and our micronutrients like iron. And then down at D here, we have our dark reaction, which doesn't necessarily have to happen in the nighttime. It's constantly happening, but that's what sustains reactive oxygen species levels throughout the night, which is why we call it a dark reaction. So really just what's happening here is reactive oxygen species are just being produced either through extracellular or intracellular production methods. So focusing on that a little bit first, we can see that these cellular organelles here are producing a lot of hydrogen peroxide and then pumping that into the surroundings. But what I really want to focus on is this extracellular reaction sequence happening here. So you really see that molecular oxygen, O2, is interacting with these extracellular organelles and undergoing either a 1 or a 2 electron reduction to produce superoxide and hydrogen peroxide respectively. And then our superoxide is then being reduced further into hydrogen peroxide pretty rapidly. So why we really care about reactive oxygen species and the way they interact with our nutrients is that they act as a reduction agent for our nutrients and have this clear correlation here. So in this graph, we see on the x-axis time in seconds, and on the y-axis we have our concentrations of iron 2 and hydrogen peroxide in nanomolar per liter. So the red line is representing our hydrogen peroxide, and as we can see, that is increasing steadily until we reach a new steady state equilibrium condition. And as that is increasing, we see this definite decrease happening with our iron 2 until that reaches a new equilibrium with the hydrogen peroxide. So why we really care about our iron cycling is iron is a limiting nutrient in the water column for a lot of things. One of those is our growth rate within phytoplankton. So this graph is showing P. carterae, which is a type of phytoplankton. And as we see increases on our log scale of iron concentrations, there is this increase in growth rate occurring. So some environmental implications for high concentrations of reactive oxygen species. We have reduced micronutrient availability, which, as I said, that limits growth rate. It also limits productivity. So this graph here, we have concentrations of hydrogen peroxide on the x-axis in nanomolar, and then on the y-axis, we have percent production inhibition. So a little bit counterintuitive. We have an increase in inhibition happening as we have our increase in hydrogen peroxide. So we're going from 50 to 500 nanomolar and see this definite increase in inhibitions of productivity. So that low productivity is going to lead to a number of implications, which one would be a reduced CO2 uptake, which ultimately can lead into a negative feedback loop with global warming, as our phytoplankton are a main sink for CO2 in our oceans. And we also would have a reduced oxygen production happening, which can lead to an anoxic ocean and cause a lot of issues for marine life. High rates of production of reactive oxygen species can also lead to a toxic environment. So these reactive oxygen species are interacting with biological organisms such as phytoplankton, bacterioplankton, and are able to be absorbed into their cells. In the absence of superoxide dismutase and catalase, this can cause a lot of issues, which include denaturing of their proteins, DNA, and lipids. And when hydrogen peroxide and superoxide are present in high enough quantities, this damage to their cells can end up leading to a lot of marine life death. This is kind of what we see what happens when we have HABs, which are harmful algal blooms occurring, because one thing that HABs are really good at doing is producing hydrogen peroxide, and that is one of the main reasons why we see massive fish loss when we have HABs occurring. So moving into some previous work, that's the basis of my model. We base it around the Fawn and Dune 2011 model, which assumes steady-state iron and steady-state solar irradiation. We had to take into account that superoxide has a very short half-life on the order of seconds. So it's really hard to constrain that. And one way we attempt to constrain it is through our hydrogen peroxide concentrations. So another thing that we had to take into account is that the biochemical sources and sinks of peroxide are more important than the photochemical processes. So what we expect to see with our model is not a clear dependence on photolytic processes, because this biological input is really dominating the production pathway. And then we also see in the never-ending battle between kinetics and thermodynamics, Allison, we have kinetics winning here. So because of the biochemical reaction sequences happening, the system is driven far from thermodynamic equilibriums, so we really have to rely on kinetic rate constants to produce our model. And then finally, we had to take into account the high level of inorganic iron that's maintained overnight is due to interactions with reactive oxygen species in that dark reaction sequence that I showed you before. So some of the goals of my model, as well as the reaction series, just to orient you to how all of this works. So the first goal of our model is to expand on the findings of Fawn and Dune's 2011 study, as well as Fawn's 2008 study. We hope to do this by determining the effects of reactive oxygen species on the iron cycle, determining the impact of biological and photic effects on the iron cycle and reactive oxygen species. And then ultimately, we hope to determine the effects of reactive oxygen species and iron cycling on ecosystem function and biodiversity. So the parameters of my model, I coded this whole thing in MATLAB. It's centered around 32 degrees north, which is represented by this red line up here on the map. There's really nothing special about 32 degrees north, other than that's where I found a lot of data, so that's why I based it around that area. We assumed solar equinox for our light variations, which is really just a fancy way of saying we have 12 hours of day and 12 hours of night with maximums of our solar radiation occurring at 1200 hours. We really just did this for simplicity's sake and hope to change this at later parts of our data analysis. We also assume a homogeneous mix layer, which just means that we have even amounts of our reactants throughout the mixed layer mixed evenly. And we assume that our mixed layer has a depth of 60 meters. And we also assume that we have an open system because we have this dust deposition occurring, which dust has a large amount of iron in it, which is our main source of iron in the oceans. We also have this sinking occurring of our particulate iron, and we're assuming that there's no mixing occurring between our mixed layer and ferrocline. So once it's gone, it's gone. It's not being recirculated, which this is where our model really departs from classical physics, because physical oceanography will tell you that that's not what's happening. We have these clines happening in our oceans, and everything's just getting circled back up. And then finally, we have variations with our steady state and variable states of our iron and our light inputs, which you'll see in a little bit what that really means. So this is really the basis of my model here. I really relied on this K value, which is our kinetic rate constant. But instead of having you look at this, because this is confusing to look at and doesn't tell you a whole lot, I went ahead and made a chemical reaction map, which again can still look kind of daunting, I get that. But for environmental chemistry, this is really our bread and butter, and this is what helps us explain everything. So really to take you through it, we have these light-dependent reactions occurring at the top here. Everything that has an H nu on it, that is just a photon coming in and interacting with those molecules to produce different variations of our chemicals here. And then really, I just tried to find ways to link our superoxide and hydrogen peroxide terms into our iron cycling and just show how all these interactions are happening. So moving into the results and discussions of my model, I'm going to start off with dissolved organic iron reactions. So dissolved organic iron is everything in red in this. So it's just our iron 2, our iron 3, and our iron ligands. So you can see at the top here, we have our iron 3 and our iron ligands being photolysed into iron 2, which then is reduced into iron 3, which then interacts with ligands and forms an iron ligand, which then cycles back into iron 2, or is photolysed into iron 2. And the cycle just keeps going back and forth here. So this is really the basis of my model just ended up outputting all of these graphs. So I had 4 different conditions that I really focused on here. So the green line is our steady state iron and variable light conditions. The blue line is our variable iron and our variable light conditions. The black line is our variable iron and steady state light conditions. And the red line is our steady state iron and steady state light conditions. And on the x-axis here, we have time in hours. And then on the y-axis, we have total dissolved iron in nanomolar. And total dissolved iron is just the summation of our iron 2, iron 3, and iron ligands. So as you can see here, we have in our green line, our steady state iron variable light. There's these sudden transitionings happening here with maximums occurring during the night and minimums occurring during the day with almost instantaneous transitions between the maximum and minimum and steady states achieved during the day and during the night. So for our blue line, we have our variable iron and variable light condition. And this is showing a little bit more gradual transitionings occurring with, again, maximums during the night, minimums during the day. But instead of this instantaneous reaction happening, we have more of a long-term reaction happening over the course of about 2 hours, with steady states occurring during the day, but a steady state never being reached during the night, with the concentrations constantly increasing over the night. And then our variable iron and our steady state light conditions, we just end up reaching our steady state condition almost instantaneously, and that just is maintained, which makes sense because we're not varying our light reactions. So breaking this into its components, we have our iron ligand, which you can see there's a really good correlation here, and our iron ligand is really dominating our dissolved iron term, with just slight variations in the maximum and minimums occurring. We have just a very slight difference in our maximum here, and then it's a little bit more noticeable within our minimum. And that's really just explained by our iron 2 and our iron 3 terms. So our iron 2 term, we have maximums happening during the day and minimums occurring during the night. And we show this clear dependence of our iron 2 term on solar radiation levels, because we see our maximum occurring at 1200 hours, which is when our maximum is for our solar irradiation term. And then for iron 3, we see something really similar, with maximums occurring at 1200 hours and minimums occurring at night, and again with steady states occurring during the night. And the steady state happening at night is really explained by our dark reaction sequences happening with our reactive oxygen species, which are able to turn our iron ligand back into iron 2, which then cycles into iron 3. So comparing our data to that of Fahn's 2008 study, we see this discrepancy here. We have much larger numbers than that of Fahn's 2008 findings, with our maximums in the scale of nanomolars and his in the scale of picomolar. So one reason why I think this is happening is the lack of a scavenger term for our iron. So realistically, what would be happening here is there would be another output of iron into biological uptake by phytoplankton and bacterioplankton. And I think that by incorporating this term in here, we would be able to sufficiently lower our iron concentrations into the range that Fahn 2008 found. Moving into another form of iron, we have colloidal iron, which is a little bit simpler to look at. We have our colloidal iron in red here being photolized into iron 2, which then is reduced into iron 3, and then cycled back into colloidal iron. And we can see that there's this oscillation happening with our colloidal iron, which can be explained by our solar radiation, but there's not a clear dependence on our solar irradiation because the maximums of our colloidal iron are happening around 1600 hours and minimums occurring around 0600 hours. And this can really be explained by this cycling happening here, where iron 3, which is produced due to high levels of iron 2 during the day, so this is like really what's explaining how we're getting this little bit of a lag happening here, because our iron 3 is a source of colloidal iron, but it's also a sink for our iron 2. Our particulate iron reaction map is pretty similar to that of our colloidal iron, with particulate iron in red being photolysed into iron 2, which is reduced into iron 3, and then back into particulate iron. But we also have the sinking term happening here because particulate iron is heavy, and it's also able to interact with other molecules in the water column to then continually add to it and make it heavier and allow it to sink out and deposit into lower layers of our ocean. So much like our colloidal iron, we see this oscillation happening with our particulate iron, with maximums occurring at 0600 hours now and minimums occurring at 1600 hours. But there is this like really small variance happening here with our maximums of particulate iron occurring around 4.96 micromolar and minimums occurring around 4.958 micromolar. So that's telling us that this variance is actually really pretty insignificant. So we can really just follow our steady state light conditions in this instance, which do not trend towards a equilibrium here. You can see that they're slowly decreasing. And I think that this can be explained by a discrepancy between our dust deposition term and our sinking term, with our sinking term being a little bit faster than our dust deposition. Okay, so moving into our reactive oxygen species, we have our superoxide reaction map, which shows a biological input into our superoxide, as well as the photolysis of CDOM, color-dissolved organic matter, as well as the photolysis of molecular oxygen. And then there is also a reduction of superoxide into hydrogen peroxide. And then there's all these reactions happening with our iron. So superoxide is showing a clear dependence on light reactions, which is pretty unsurprising, as there are all those photolytic terms feeding into it. And superoxide showing, again, maximums at 1200 hours and minimums during the night that are steady states. And they're steady state because of the constant input from biological factors. So we can see that there's this discrepancy between that of Fahn's 2008 study and our model. So we ended up having concentrations much lower than that of Fahn 2008's for superoxide. This can be explained by the interaction between iron and superoxide. Because we have elevated levels of iron in our system, we end up having reduced concentrations of superoxide. Hydrogen peroxide is a little bit simpler of a map. We have our biological input, photolysis of CDOM, and then reduction of superoxide feeding into it. And then there's this interaction with iron 2 and iron 3. So we see an oscillation happening with our hydrogen peroxide, with minimums occurring around 1600 hours and maximums occurring around 0600 hours. So there is a slight dependence of our hydrogen peroxide on photolytic processes, but because of the interactions with biological inputs, we don't have a clear dependence on photolytic processes alone. So comparing that to our Fahn 2008 study, we can see that we kind of ended up with the inverse of what Fahn and Dune showed, with our maximum being around 0600 hours and theirs being at 1600 hours, and our minimum occurring around 1600 hours and theirs occurring around 0600 hours. we also ended up with elevated levels of hydrogen peroxide in comparison. So these discrepancies can really be explained within our biological input and our CDOM input, because our term here that we used, I ended up oversimplifying like the reaction map just to show you, but we ended up having this coupling of our phytoplankton and our CDOM terms with our production of superoxide, assuming that superoxide transforms rapidly into hydrogen peroxide. But reviewing other papers, we can see that phytoplankton and CDOM can also produce hydrogen peroxide directly and not just indirectly. So by finding some more terms to add in for those, we would be able to hopefully flip our model to be able to show our minimums occurring at 0600 and maximums occurring around 1600, like that of FON 2008 study. And then our levels of hydrogen peroxide are elevated, primarily due to iron again. So this iron term interacting with superoxide, producing hydrogen peroxide, is why we would expect to see elevated levels of hydrogen peroxide in this model. So in conclusion, so our iron ligands are dominating the dissolved organic iron concentrations in our model. Iron 2 is much more abundant than iron 3, which is preferable for biological uptake, as iron 2 is less reactive than iron 3. Iron concentrations are elevated due to a lack of scavenger terms and affect our reactive oxygen species concentrations. And hydrogen peroxide is decreasing during the day due to a lack of direct CDOM production and biological production terms. Some future work to help correct these discrepancies, as well as further our study, would be to incorporate different literature values for CDOM and phytoplankton productions of peroxide, add a cellular uptake variable incorporating different species of phytoplankton and bacterioplankton, uptake rates for iron, incorporate variable dust depositions to add a level of seasonality. As wind patterns change, dust deposition terms would also change. Add a variable precipitation into this, as reactive oxygen species are deposited within precipitation. So that is a main source of reactive oxygen species. And as climate change takes effect, we will expect to see changes in precipitation patterns around the world. We would also like to add a variable pH term into this, as reactive oxygen species production is increased under low pH conditions. And as the ocean uptakes more and more carbon dioxide, it becomes more acidic, which would lead to higher levels of reactive oxygen species production. And finally, we would like to increase our time step to a magnitude of hours. Currently, it's sitting on the scale of seconds, and as we try and increase it, it has been failing. So hopefully by tweaking a few bugs, we would be able to increase this to a time scale of hours and start to look at more seasonalities occurring within the model. So I'd just like to acknowledge the Noah Holling Scholarship for funding all of my research this past summer, the Geophysical Fluid Dynamics Laboratory for hosting me, Dr. John Doon for being my mentor over the summer, Dr. Deanna Donahue for being my advisor here and helping me to even find this internship in the first place, Dr. Rachel Dowdy and my classmates for helping me edit all of my slides and just really make this a coherent presentation. So then there's my works cited. And then any questions? Cool. Just hit the mute thing. Yeah? You can hit that. Okay, cool.
All right, thank you, Justin. Justin would be happy to take questions at this time. Chris.
So I have a question about your time scale of your model. So you're assuming that 12 hours a day, 12 hours a night. So what if you have some kind of like different night and day ratio. For example, like extreme at the pole is going to be 24 hours a day or 24 hours of night. What would the peak be like then?
So I think that is when we would start to, if we were to just have an extreme of 24 hours of dark or 24 hours of light, that is when we would get into those steady state light models, which would be able to show that they just end up going to steady state conditions almost immediately, and we just have this kind of straight line of concentrations happening. I can like show you an example of that real quick. Sorry. That button, sorry. Yeah, so like this red and this black line in here, those are those steady state light reactions that are happening. So that's just assuming like basically either a 1 or a 0 for the light reactions. So all of these light terms in here would just not have any variance happening.
All right, thank you.
Yep.
Carmen?
The last slide that you had, like, I forgot the number already, but were you, yeah, the last presentation slide, you had mentioned that you're trying to up the timings, but it kept failing. And I was just wondering why.
So there must be like a bug or something within my code. I think it also might have to do with our superoxide term just because of how rapid that reaction is happening. So maybe finding better ways of confining that reaction would help us.
Allison?
I was curious about what the source of the rate constants are. So in terms of like, are those experimentally determined?
Yeah, so a lot of those are the result of mesocolumn experiments around 32 degrees north. So essentially they just have like a kind of like a controlled environment within the ocean. So
yeah,
like,
that's what I'm curious
yeah.
about. Like what variability is there within the rate constants as you're using as your input?
There's definitely some variability there, and it's definitely going to vary within location. So that is where like trying to build out a complete global model would become a little bit more difficult because we would have to be sourcing all of these rate constants.
Cool.
I have a question. I want to ask about the seasonality. You said that the rate of deposition of dust.
Yes.
What season would we expect higher deposition of dust? Is that a summer thing?
It really depends on where you're at within the world. So based on how it really goes into a lot of atmospheric chemistry and physics. So basically, like based on wind patterns, based on like if it's an El Nino year, La Nina year, you're going to see differences in wind patterns and strengths. So dust that could be coming in all the way from the Sahara in some years might not be, depending on how those trade wind interactions are happening.
Other questions for Justin? Eder?
You said that you had ligands bind to iron, right?
Sorry, what?
My fault saying. You said you had ligands bind to iron?
Yeah.
Do you know what type?
That's a good question. There's a lot of different ligands that could potentially bond with iron. So this was really just kind of like a generalized ligand term that we used. Yeah, I don't really have much of a better explanation than that.
You're all good.
Other questions? Let me check the Zoom chat one more time here. Nothing but admiration for Justin in the Zoom chat. So we'll call it. So our next speaker is going to start at 4 o'clock. So if you're interested in hearing Matthew's talk, hang out, have a snack, talk to your neighbor, and otherwise, thanks for joining us.